Hello student, the name of the topic which is going to be discussed is Trypanosoma brucei gambiense, its structure, life history and pathogenicity. Trypanosoma brucei gambiense is an protozoan endoparasite human strain, live in the blood, then spleen, the lymphatic glands and certain others part of the cerebrospinal fluid, and it is also carried by one important insect species which is commonly known as tsetse fly. Scientifically it is known as Glossina palpalis. This parasite causes a disease called African trypanosomiasis, commonly known as sleeping sickness, and it is mainly found in the western and central parts of the Africa and some other tropical countries. Let me explain about the structure of the Trypanosoma brucei gambiense. Actually it is a slender, elongated and microscopic body, and it contains so many cytoplasmic granules and some other cell organelles like endoplasmic reticulum, Golgi bodies, and mitochondria and nucleus. So the body is surrounded by thin elastic firm pellicle. The pellicle maintains the general shape of the body, and the pellicle is extended in at one side of the body in the form of a very membranous fall, and this membranous fall is known as undulating membrane. A single flagellum is present which is uniflagellate. The flagellum arises from the basal granule situated at the posterior most part of the body. The flagellum runs forward and attaches along the sides of the body and runs upward, and it forms the boundary of undulating membrane. Now let me come to the another important polymorphic forms of Trypanosoma brucei gambiense. There are three forms. One is A mastigote, which is less manual, and epimastigote, and which is critidial, and promastigote is a leptomonad, and trypanosome, trypomastigote. Let me explain about the polymorphic forms. Less manual or amastigote. It is a oval or rounded structure having a nucleus at the center, and basal granule and a kinetoplast is located just in front of the nucleus, and there is no say undulating membrane, and the flagellum is just in the form of a fiber-like structure. Then the second one is promastigote, leptomonad. The body is elongated and the nucleus is at the center, and basal granule and kinetoplast lies at the anterior most part of the body, and from that the flagellum arises, and it has no say well developed undulating membrane. Then the next one is the critidial or epimastigote, and it is it has basal granule and kinetoplast just in front of the nucleus, and from kinetoplast and basal granule the flagellum arises, and it runs upward and hangs freely and the anterior most part of the body. And the last one is trypanosome, which is also known as trypomastigote, and it has also some features, and the body is slender and elongated, and it has some other features like basal granules, kinetoplast, and these things are located at the posterior most part of the body, and from basal granule the flagellum arises and runs in a long way towards the anterior most part of the body, and opens freely that particular flagellum and hangs as a free flagellum. Next, let me explain you about the life history of Trypanosoma brucei gambiense. Life history is completed in two hosts. The first one is primary host, that is the main. Secondary or intermediate host, that is the blood-sucking fly, scientifically known as Glossina palpalis, commonly known as tsetse fly. So it consists of two parts. One part is life cycle in man. So when the infected fly bites a mane and it inoculates the particular parasite into the body of the mane, then they make their way into the cerebrospinal fluid, and then it remains some more time in this cerebrospinal fluid, and in the meantime some fever-like condition is developed, and that kind of fever is known as gambia fever. Then when this parasite reaches the cerebrospinal fluid, some unwanted changes occur in the body of the patient, and say some conditions like lethargic conditions is leading to a lethargic condition, and the patient feels very inactive. So it has given the name sleeping sickness to the disease. Then the next part is this parasite is multiplied by means of certain other methods, say longitudinal binary fission. By means of this, it increases their number, and it forms three types of structure: long and slender forms with a free flagellum, and the second one is the short and stumpy forms with reduced flagellum, and the third one is the intermediate forms. The parasite increase and decrease their number in the blood of the mane. During the period of decrease, the short and stumpy forms is having the capacity of retaining in the body of the patient, and during the period of the depression, and it can be very more powerful, and it is capable of development in the body of the insect, that is tsetse fly, commonly known as Glossina palpalis. Then the next part is life cycle in tsetse fly. When the tsetse fly sucks the blood of an infected man, the parasite enters the body of the fly, that is tsetse fly, and it remains some more time in the midgut of the fly, and for some other days. Then it starts increasing their number by means of say one method, that is the longitudinal binary method, and it increases their number enormously. So then after about 10 to 15 days, this parasite is in the form of slender, long and slender body, and develops in a large number, and it can be transformed into the some other part, and it remain in the anterior part of the say stomach of the particular fly. That anterior part of the stomach is the proventriculus, commonly known as proventriculus. Then the trypanosome, that is the particular parasite, make their way towards the salivary gland of the fly, and then it remain attached to the inner wall of the salivary gland, and then it will remain some more time in that particular area. At this stage, the parasite increases their number enormously by means of certain other say methods, longitudinal and binary fission, and it can be transformed into the critidial forms. The critidial forms is characterized by shorter flagellum and very shorter undulating membrane, and the development of the critidial continues for about 2 to 5 days. Then it will be transformed into a metacyclic form, which is commonly known as trypanosomes, and which is now infective in the infective stage. The metacyclic form passed down through the salivary duct of the salivary gland and hypopharynx. When the parasite sucks the blood of the infected person, automatically the metacyclic forms enter into the body of the man along with the saliva of the fly, that is the tsetse fly, Glossina palpalis. So the whole cycle in the tsetse fly is usually takes about 2 to 30 days. Now let me come to the pathogenicity. The bite of an infected fly is followed by say some abnormal condition in the body of the patient, and which is developed the itching and the irritation near the wound, and then frequently dark red lesions develop in the body of the patient, and in the meantime, say the parasites increase their number in the blood and absorbs all the nutrients from the blood, and it leads to the condition of the anemia. This is also another pathological effect caused by this parasite. Then in addition to this, certain other changes have also taken place. Then fever, headache develop, accompanied with the loss of weight, then say joint pains, and some other unwanted changes. Then the parasite attacks the lymphatic glands, resulting the swelling of the glands, and then they enter into the cerebrospinal fluid and the brain, causing a sleeping sickness like condition. That is what we call the sleeping sickness to the disease. Say another important pathological effects caused by this parasite is the development of lethargic conditions and the recurrence of fever due to the infection of this parasite. So with this, I conclude this discussion at this stage. Say, dear students, if you have any doubt or queries regarding this topic, and you may link through this email which is given below.